enzymes are very sensitive to conditions, and there are a number of factors which can affect their function, such as changes in temperature, changes in pH, the concentration of substrates, the concentration of products, and other substances that may compete with the substrate for an active site. This video addresses the effect that changes in temperature and pH have on enzymes. Now, in order to understand how temperature and pH have an effect on enzyme activity, you have to recall that enzymes are proteins. Proteins are chains of amino acids that are joined together to form polypeptides. And a polypeptide is only called a protein once it folds into its functional shape. Proteins form their shape due to hydrogen bonds that form between the amino acids and interactions between the R groups. And the R groups are the side chain of the amino acids that make them unique from other amino acids. As a result of this, they form a specific shape referred to as a tertiary structure. When there is an interaction between multiple tertiary structure proteins, you end up with a quaternary structure. These tertiary and quaternary structures are critical for enzymes to function properly. The shape is specific to the substrate they bind with in order to catalyze a reaction. Therefore, if the shape is altered in any way, it affects how well the enzyme can catalyze a reaction. Enzymes exist in different cellular environments, and each enzyme is suited to function at an optimum level within the specific environment that they are located. When enzymes are not in environments that suit them the best, it affects their functionality, and therefore they don't work at their optimum level. An example of this is temperature. Enzymes are affected by temperature, and they have an optimal range in which they operate. Enzymes operate best in the temperature of the environment in which they are found, and this can vary depending on where the enzyme is found. For example, an enzyme from a human would be expected to operate at an optimum level at a temperature of around about 37 degrees. So this is the optimal temperature. Because this is the temperature or the core body temperature of humans, whereas an enzyme from a bacteria that lives in a hot spring would be expected to operate at a temperature of around about 73 degrees Celsius. Because this is the temperature of the water in the hot springs. This does not mean that these are the only temperatures in which these enzymes will catalyze reactions. And you can see from the graph here that enzymes from humans, in this particular example, will operate in a range anywhere from about 26 degrees to about 41 degrees. But you'll notice that it doesn't operate at quite the same rate of reaction as it does at 37 degrees. So it decreases in rate of reaction as you decrease the temperature, and also decreases in rate of reaction a little more rapidly as you go over the optimum temperature. And similarly, if you deviate either side of 73 degrees for the enzyme from a hot springs bacterium, it also reduces the rate of reaction or how well that bacteria enzyme functions. So how do changes in temperature affect enzyme activity? Well, enzyme activity generally speeds up as the temperature climbs because the reactants have more kinetic energy at high temperatures. Increased kinetic energy means that molecules become more active and collide more often. So if you increase the collisions between the enzyme and substrates, the rate of reactions is going to increase. So increase in temperature results in an increase in kinetic energy, which means more collisions are occurring. And you can see that happening on this graph here for both the human enzyme and the bacterium enzyme. You can see how an increase in temperature from approximately 55 degrees up towards 73 degrees is resulting in a faster rate of reaction. And this is simply because you are increasing the kinetic energy and you're getting more collisions as you head towards your optimum level. But you'll notice after the optimal temperature for both of these enzymes that there is a rapid decrease in the rate of reaction. And this is because when you add too much kinetic energy to your system, that you're actually affecting the shape of the enzyme. You're altering the shape of the enzyme and therefore affecting its functionality. Now, if an enzyme is heated up too much, its shape changes enough to break the bonds between connecting units of amino acids, and the enzyme can no longer return to its original shape. In this case, it is destroyed. However, enzymes can return to their functional shape if they are heated to temperatures that are slightly over the optimum temperature, and then they can reshape or return to their original shape. And this is referred to as renaturation. So this image here shows this process exactly. So a normal protein functioning within a range that is fine for that particular enzyme has a very specific shape. And then, of course, after a certain temperature, if you heat it up too much kinetic energy breaks a lot of the bonds in that enzyme and it ends up unwinding, and that's referred to as being denatured. Now, if you don't heat it up too much, you can, of course, then renature. It can reshape. If you heat it up beyond a point where you can no longer repair it, that enzyme is irreversibly damaged. pH is another factor that affects the activity of enzymes. Similar to temperature, enzymes exist in environments that have a particular pH, and the pH of the solutions surrounding an enzyme could be acidic, basic, or neutral. Each enzyme has an optimal pH at which it works. Some enzymes can work in a broad range of pH environments, while others are very sensitive and will only work in a narrow range. So pepsin, for example, is an enzyme that works with an optimum pH of around about 2. But if you deviate to a pH slightly more or less than that, it doesn't function very well. So it has quite a narrow range of pH in which it will function. Amylase is a enzyme that you can find in your saliva, and you can see that it operates at an entirely different pH. In fact, its optimum pH is around about 6.5 to 7, and that's because that is the pH of the environment in your mouth. And that has a slightly wider range in which it will operate. Now, arginase operates at a pH of around about 9.5 and has a much broader range in which it can operate. Changes in the optimal pH environment for an enzyme break down some of the bonds in the enzyme, changing its shape and therefore its effectiveness to function properly. This change in the shape also affects its active site, so much that the enzyme becomes denatured and can no longer catalyze a reaction. A change in pH can also have an effect on the substrate, causing it to no longer fit into the active site, meaning that the enzyme becomes less effective simply because the substrate can no longer fit.